find these slides?
They're posted on Canvas
Okay.
under the exam 2 stuff.
Oh, I should probably make an announcement now, you think?
Yeah.
Okay. Just like
Or not exam 2, it's final exam stuff.
that it's open.
Yeah. You guys can all see the work and process of making an announcement through Canvas. Congratulations. Now, yes.
Yeah. Kelly, you want to help him
Yeah,
with
yeah,
that?
yeah. What group are you in?
Where'd you get that shirt, Ricky?
From, it's like it's a program with a week before classes start.
Oh,
And I was
yeah.
part of it.
Nice.
As a freshman, like,
The one credit
this
thing?
year, and then I was a mentor
Nice.
this year.
Nice.
You get a shirt for free. What? No, you get free.
It's been published.
Good job. Good job.
Thanks. Thank you.
Okay. Shall we?
Time to start,
Shall
yeah.
we begin? I don't know.
I don't have any speaker notes for mine.
I don't think I do, but I'll leave it up. I'll just leave it here just in case. Okay. What time is it?
It's one.
It is 1. Okay. Hi everyone. Welcome to the second final exam review. Yeah. How are you guys all feeling about tomorrow's exam? Like, okay or mixed?
Thumbs up,
Thumbs
thumbs
middle,
down.
down.
Middle, okay.
Okay.
Not bad.
Mostly
Yeah.
decent. Better than I've seen in years past, so that's good. Yeah, so just stop me at any point if you have questions throughout. We'll be going through topics 7 through 10. Yeah.
I just feel like there's like so much information that I'm not sure we're just gonna ask.
Yeah.
Do you know like questions that she'll probably ask like 100%? Like probably like the trip offer on, and then like I don't know what else. Like,
Yeah.
you think.
Let's see. We have some tips towards the end too, but for sure there will be something trip opera on. There will be cell cycle stuff. She'll probably combine topic 7 and 8 and be like, this is happening in the cell cycle, like what stage of the cell cycle is it in, stuff like that.
Also, like mutations in any of the proteins involved in DNA synthesis, cell cycle, what else? Like replication, anything like.
Yeah.
Yeah.
What was topic 3?
I actually don't have no clue.
I think that was
CDK
What was
pathways?
topic 3?
Is that
It's
one?
before trip opera.
that's like Randgap.
Oh, yeah,
Rangap.
yeah.
Oh, yeah, yeah. Import,
Yeah, you might
export.
have some
Yep.
nuclear import-export stuff.
Sure.
Yeah.
Just think of it as like, okay, list out the proteins involved in that topic. What happens if this one gets mutated?
Yeah.
Gain of function, loss of function. That's like the very like fundamental
Yeah.
way of
The
questions.
majority of your questions are going to be on what happens if if something happens to this protein, or what kind of protein is this? Is this an ATPase? Is this a GTPase? Stuff like that. So, yeah. Did you start a Zoom recording?
I did an OBS thing,
Okay.
yeah.
All right. Any other questions before we start? Okay. And then we also have quizzes, exams, all your stuff from the previous semester. You can pick that up at the end if you didn't already grab it. So yeah. Okay. So we'll start with topic 7, cell cycle 1. So this was all the way back from exam 2. So this is the least recent stuff that you've known. So yeah. We'll just start with a quick question from stuff. This was on not this year's exam 2, last year's exam 2. But yeah, so make sure, take a minute to do this, I guess. Just write that down. Who's calling me right now?
Who's calling my phone?
Do not call my phone. Do not call my phone.
Don't call my phone. Don't come by my house.
We're done.
I hate these anti-dust ones. They write so.
Are they actually anti-dust?
Yeah, it's like it's good, but like it makes worse noises. Actually, it's not that bad.
That's not that bad.
I guess it's because. Maybe it is the chalk ones that I don't like. The other ones, bad ones. Whatever.
Okay, I'll just cut it off there because we have a lot of stuff to get through today. So just quick basic overview before we get into the more detailed stuff. First, you have pre-RC formation because, of course, you have to have low levels of CDK in order to form the pre-RC. Then you would have G1 cyclin CDK activation, E2 activation, which is phosphorylation of RB by G1 CDK, replosome formation on origins of replication, DNA replication, the G2 checkpoint, the spindle assembly checkpoint, and then finally cytokinesis, which is when the cell splits into 2. Yeah, any questions on that? Yeah.
Can you like just quickly go over like the represent formation?
Yeah, I'll go over that in more depth in like a couple slides if you want to. Yeah. Okay, so we'll just start with the phase of the cell cycle. So G1, this is a growth phase. Cells receive a growth factor signal, check for DNA damage, size, and nutrient requirements before committing to cell division. This is where RB is phosphorylated, leading to the release of E2F. S phase is when DNA replication occurs. That starts the moment DNA replication occurs. G2 phase is another growing phase, getting prepared for mitosis, which is M phase, which is when the cells actually divide. And then G0 is the phase the cell is in most of the time. No replication happening. Often, if something goes wrong in the cell cycle or something happens in a cell cycle checkpoint, if the cell isn't committing apoptosis, it's instead just reverting back to G0. And the cell cycle is controlled by CDK, which is cyclin-dependent kinase. This is a serine-threonine kinase, which means it attaches phosphate groups to the amino acids serine and threonine, which have OH groups or hydroxyl groups on their R group. CDK is regulated in 3 different ways. So CDK has an activation loop that needs to be pulled out of the active site by cyclin. So binding to cyclin pulls that activation loop out of the active site so that CDK can actually phosphorylate its targets. But cyclin binding is not enough for CDK to gain full activity. You also need phosphorylation of that activation loop by CAK, which is CDK activating kinase. And then also Wee1 kinase can add an inhibitory phosphate onto CDK and keep it from performing its activity. So Laura always likes to say CDK is the CEO of the cell cycle, right? So it regulates everything in the cell cycle, either directly by phosphorylating certain targets or phosphorylating some other targets that might, and like priming them for different actions. think that's about it for that. There's different cyclins for each stage of the cell cycles. There's M cyclins, G1 cyclins, S cyclins, stuff like that. For the purpose of this class, there's just one CDK that binds to all of these. Yeah. So here's this again. Different cyclins help CDK target different substrates. So in G1 phase, you have G1 cyclins that are being upregulated and G1S cyclins. When E2F is activated, it helps with the transcription of G1S cyclins and S cyclins. M cyclins levels slowly rise during G2 phase, and they peak at the G2M transition point, and they get degraded by APCC, which we talk about later. Yeah, that's about it. And again, just remember, CDK bound to cyclin is not fully active. You need the CDK activating kinase to phosphorylate CDK2. And yeah. Oh yeah, and then activation or activity of CDK can also be controlled by degradation of cyclins or by the binding of CDK or CKIs, which are CDK cyclin inhibitory like molecules that bind both the CDK and the cyclin and keep it from being active. So yeah, we'll just do another quick question. This is from your quiz 7. So where in the cell cycle are these cells? Someone can just shout it out if they want to do that so we can move quickly, or we can take a minute. It's up to you guys. Okay, we'll take a minute. All right, so this first one is going to be after early G1 at any point. So CDK inhibitors like P21 are expressed when P53 detects DNA damage, and so that will bind to the cyclin CDK complex. So this can happen at any point in the cell cycle, although it'll most likely be happening during G1 or S phase, because hopefully if you're in G2 and M phase, you should not have DNA damage. And then if pre-RCs are forming on the origins of replication, that would be an early G1 because CDK phosphorylates origins of replications and keeps them from reforming during S phase. So these form when CDK activity is low, and it's lowest in early G1. Yeah, so I guess talking more about the P21 and the P53 pathway, the various checkpoints throughout the cell cycle are checking for nutrients and growth in G1, always checking for DNA damage, checking for completed replication at the start of G2 or after the end of S phase, and then proper spindle assembly in M phase. The DNA damage checkpoint is regulated by P53. So if P53 detects DNA damage, it is phosphorylated, and that keeps it from being degraded by proteins. And so once you have an active P53, it will bind to the P21 gene, so it'll bind to the actual DNA and act as a transcription factor to synthesize P21 mRNA. That P21 mRNA can then be translated into the P21 protein, which is a CDK inhibitor protein, which binds G1S CDK and S CDKs, basically preventing them from progressing through the cell cycle, right? Because we don't want to divide and grow and make more cells if we have damaged DNA, because that wouldn't make any sense. And that's how you get mutations and all sorts of fun stuff. So, yeah. Any questions on P53, any of that? I feel like that's okay. Cool. So then here's just another overview of the checkpoints. So in G1, you have your damaged DNA checkpoint, and checking for proper growth and cellular environment, making sure the cell actually wants to commit to the cell cycle, because of course this takes a lot of resources. So if you don't have the proper growth in G1 or something's going wrong, you don't want to commit to G1. Because remember, at the start checkpoint, once you've committed to the cell cycle, you're committed to the cell cycle. In S phase, you're checking for damaged or incompletely replicated DNA. In G2 phase, you're also checking for this. And then in M phase, you check to see if the chromosomes are properly attached to mitotic spindles so they can separate into separate sides of the cell and split. So now we'll just go over some the main G1 events that we need to know for this class. So we have a growth factor molecule binds. This whole cascade happens and causes G1 cyclins to be made. And so cyclins can then bind to CDK. CDK can then phosphorylate RB in two places. RB is holding E2F inactive, and E2F is the transcription factor needed to move into the rest of the cell cycle. So once RB is phosphorylated, it releases E2F, and E2F can begin to transcribe mRNA, and then which will eventually be translated into proteins that are required for S phase. So once CDK phosphorylates RB, the cell cycle has basically been committed to, right? This is the restriction point, start, whatever you want to call it. After RB is phosphorylated, it becomes inactive. E2F moves the cell cycle along. Yeah, again, same stuff. E2F promotes the transcription of proteins required for S phase, and E2F is bound by RB until the cell cycle, or the cell is ready to move on to commit to the cell cycle.
So, wait, quick question. Where?
Oh, yeah.
Yeah. Where in the cell is this happening? This whole RB E2F situation. Yeah. And why?
Because this is happening, like RB binding to E2F is restricting like actual replication from happening. That only happens on the DNA, which is in the nucleus.
Yeah.
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 And then any idea what here would be a proto-oncogene versus a tumor suppressor?
Nice.
That's a good thing to know for this exam
like Laura
too.
question, yeah.
We love tumor suppressors. We love proto-oncogenes. Yeah.
RB would be the tumor suppressor, E2F would be the proto-oncogene.
Exactly. A proto-oncogene is something that promotes uncontrolled cell division or cancer. And so E2F would be a proto-oncogene, and then RB is that tumor suppressor that's keeping E2F from being active until it actually needs to be. If you have something go wrong with RB, then all of a sudden you have a active E2F before you need it, and then that could lead to uncontrolled cell division. The other thing that we talk about in G1 is pre-RC formation. So this forms when CDK activity is low, your origin replication complex will be on your origin of replication, which we'll talk about in topic 8 and 9. ORC recruits CDC6 to the origin. And once CDC6 binds, it can recruit CDT1, which brings along two MCM proteins. Does anybody know what the two MCM complexes actually are?
Helicase?
Yes, exactly. So your MCMs are your helicases, which help unwind DNA for DNA replication. So all of this, again, is happening when CDK activity is low. Because when CDK activity is high, it will phosphorylate CDC6, it'll phosphorylate your MCMs, and it will phosphorylate ORC. Phosphorylating your MCMs makes them be exported out of the nucleus, so you can't do DNA replication. Phosphorylating CDC6 targets it for degradation, and phosphorylating ORC just prevents binding of these other proteins to ORC. Because, again, we don't want this pre-RC reforming when we don't want it. Multiple pre-RCs would lead to multiple copies of DNA, and we only want two for two cells. Here's just more MCM helicase. MCM is helicase. CDT1 leaves the origin once it drops off MCM. Yeah. And then pre-IC formation happens once G1 CDK is active. So G1 CDK can phosphorylate these proteins and actually recruit them to the ORC and prepare. Again, this is also happening after E2F because E2F is kind of synthesizing these proteins that are needed for S phase. So this is the pre-initiation complex, and the pre-initiation complex is forming after pre-RC when CDK activity is high. And these will just help in origin firing for DNA replication. And the start of S phase is when your first origin fires, and you start DNA replication. I got another question for you. Yeah. So would this mutation cause the cell to get stuck in the cell cycle? Yes or no? A loss of function in P53.
Who's got it? Who wants to explain?
Yeah.
No, because P53 is a cancer loss of function of P53 causes cancer
Yeah.
because it's a tumor suppressor.
Exactly. P53 is a tumor suppressor. If you lose function in P53, then it won't be able to make P21, that CKI, Jesus, that is keeping the cell from moving on in the cell cycle. So yeah, exactly. Cells would not get stuck if there was a loss of function in P53. What if there was a loss of function in E2F? Anybody that's not Lily? Yeah.
The cells get stuck at the checkpoint.
Yeah.
And then get recruited.
Yes, exactly. So cells will get stuck at the restriction point because E2F is important for moving the cell past that restriction point. So if you have a loss of function, you can't pass the restriction point. A loss of function in ORC, or the origin of replication complex. Yeah.
It would get stuck because ORC has to recruit all the other proteins to unzip DNA and stuff like that for DNA replication to happen. So it gets stuck.
Yes, exactly. Cells can't begin DNA replication if ORC has a loss of function. If it's not binding at the origin of replication, if it's not recruiting other proteins there, whatever's happening, if there's a loss of function in ORC, you don't get replication. Yeah, so that's cell cycle 1. Anybody have questions on any of that stuff before we move on to topic 8?
The second half. Yeah, what's up?
Yeah.
Is E2F just a transcription factor?
Yeah.
Yeah.
Okay.
Yeah. Anything else? Okay.
All right, part 2.
Show's turn.
This is pretty much S phase and onwards now. So general overview of what happens in S phase. I'll walk you through like the main events, but as we all know, it's the synthesis phase. So low levels of CDK are required to go from pre-RC, as Kyler said, after that. I think this was a quiz question you guys had, but like if high levels of CDK were around during the pre-RC formation, that wouldn't happen, right? Does that make sense to everybody? Okay, cool. So, I think, Keller, you explained most of this anyways, but I'll go over it again. So phosphorylation of ORC in S phase inhibits the new binding of CDC. So you don't want pre-RCs to form again in S phase. So yeah, after that, CDK phosphorylates CDC6, which leads to proteolysis. Again, I think ubiquitinylation, people are pretty familiar with that, right? Cool. And then, yeah, CDK will phosphorylate MCM and cause them to be exported, like Keller said. But not for pre-RC, but for pre-IC, you do need active CDK. So then DNA polymerase can be recruited, and then the origin of replication fires, and then you start actually replicating DNA. Is that all good with everybody? Yeah.
Did you go over that ubiquitin?
Yeah, so essentially it works by tagging something that you want to be degraded, or not you, but the cell that needs to degrade something will tag it, and that tag is the ubiquitin.
Yeah.
Yeah.
Ubiquitin is
yeah.
a protein
Yeah.
that gets attached to the proteins that need to be
And that
degraded.
gets sent to the proteasome.
Yep.
Proteasome. Yeah. All right. Cool. All right. Another question. This was also a quiz. Yeah. Would the following mutations cause cells to get stuck in the cell cycle? So mutation in CDK phosphorylation site in ORC, so that CDK can no longer phosphorylate ORC.
The cells would not get stuck. They would use the same genes and then it would cause cell death or proteolysis.
Yeah, there was like 2 potential answers to this. Yeah, what do you have?
Is it possible that if it can't get rid of ORC, then it's possible that the DNA replicates multiple times during S phase, but then would the cell catch that at the end of S
Yeah.
phase and just probably decide to end itself because there's no way to remedy that?
Yeah, I think those were two acceptable answers for the quiz. Either it won't stop and it'll just have multiple copies, or it'll recognize that it has too many copies and then just go through apoptosis.
Good question.
Yeah.
Why wouldn't CDK use phosphorylate everything else? Like MCM and CDC? Because ORC can't be phosphorylated, right?
It can be. So
Oh,
it
so
prevents
Yeah.
Which one's the mutation, CDK or ORC?
the mutation
On
is-
ORC. It's so
Yeah.
it can't get phosphorylated in this situation by CDK.
Yeah. I mean, technically, if you have, like, CDK could still phosphorylate the other proteins, right? But I don't know. The less regulatory steps you have, the worse it is, right? And so, for the purposes of this question, we're just thinking, what's going on with ORC? And so, yeah.
Oh,
If you also think about it, like if it first phosphorylates ORC, then it'll release all the other components. So if it can't phosphorylate ORC, it's gonna have a harder time binding to other stuff.
yeah.
Yeah. Yeah. So both answers would be acceptable as long as you explain out your thought process fully. So you want to say like, hey, I think if it's gonna go through apoptosis, this is why it's gonna, have multiple copies, but then get stopped at the checkpoint, right? Or if you think it won't get stopped, here's why. Like, just make sure you explain every step of thinking. That's what gets you points on these multiple choice questions. Okay, so in G2 is another growth phase. Yeah, it's another growth phase, but it's important in activating M-cyclin CDK. Why? What does active M-cyclin CDK mean? Yeah,
Start of M phase?
exactly. You start M phase. So M-cyclin is transcribed and translated in S phase because everything's transcribed in S phase, or it's replicated, but it's also transcribed then. But that doesn't mean that M-cyclin CDK gets activated immediately. It needs to be, again, phosphorylated. CDK needs to be phosphorylated by CAC, and then also the inhibitory phosphate from WEE1 needs to be taken off, and then it'll bind to cyclin, the M-cyclin, and then be active. Again, CDC25 will remove that WEE1 phosphate. There are some pathways that we talked about that have like a loop that partially activates CDC25. So that's like a positive feedback loop. Once it gets partially activated, then phosphorylation will fully activate it, and then active MCDK will that'll inactivate the WEE1 kinase as a whole.
It's on the next slide, too.
Yeah, okay. Yeah, here's the other diagram. So, as a reminder, just CDK cyclin, that's not active. The phosphate needs to be taken off, the one that was put on by WEE1, and then CAK needs to put its phosphate on. Yeah.
And these two positive feedback loops, right? So it's MCDK phosphorylating CDC25, so it can remove that inhibitory phosphate, and MCDK phosphorylating WEE1, so it can't add that phosphate anymore. Causes basically a huge surge of MCDK to be active all at once, and so you move quickly from G2 phase into M phase because these positive feedback loops are basically upregulating everything at the same time, leading to like a lot of MCDK activation quickly.
Okay, quick question for y'all. What happens when each of these three things get mutated? So gain of function in WEE1, or loss of function in CDC25, and increase in KD. This is a throwback to earlier on in the semester, but increase in KD between MCDK and CDC25. If you want, I can go back to the other diagram. This is different. Maybe 3.
Oh, fuck, I didn't change it.
Increase in KD.
You're gonna have to X out of the slides when I do, so it changes itself.
I mean,
I'm saying you might have to it updates itself. Call on Niall, make him answer something.
Give it 30 more seconds.
What time is it? Are we making good time? Oh yeah, we're making good time.
All right, who can tell me what we1 does, and then what happens when there's a gain of function? Somebody from this side? Yeah.
It adds that inhibitory phosphate.
Mm-hmm.
So with a gain of function, you just like keep having that inhibitory phosphate on it.
Yep.
So CDC25 would like remove it, just like not be inhibited.
Yeah. So what do you think would happen in terms of the broader cell cycle, or broader G2 phase?
It would like overly activate.
So would it have an easier or harder time getting into M phase?
Easier.
If we1 is more active?
Oh wait, I mean harder time.
Yeah. Yeah, so it'll have a harder time getting into M phase. What about the second one? Loss of function in CDC25. Yeah.
It would have a similar effect because CDC25 sort of removes the inhibitory phosphate.
Mm-hmm. Yeah. So harder time getting into M phase. And then increase in KD between MCDK and CDC25. When you answer this, tell me what KD is, tell me what it means, and then tell me what MCDK and CDC25 do, and then what happens if there's an increase in KD. So I want you to explain the whole 9 yards. Yeah, Ricky.
If I remember correctly, KD is the dissociation constant,
Mm-hmm.
and it's like higher KD means lower affinity, and then lower KD means higher affinity.
Yep.
So if the dissociation constant increased, that means there would be a lower affinity between MCDK and CDC25.
Yep.
So what I think that means is that it would slow down positive feedback loops. So I guess you could argue both ways. Like if it's like a big increase and it can no longer like bind to it, it would stop like the positive feedback loop and you could get stuck there, or it would just be slower than it would be normal.
Yeah, that's a perfect answer. Just a reminder, when you answer, you want to answer like that. You talk about, okay, what does this change mean? What does the normal thing happen? Like what happens in the normal situation, and now what happens in this overall change situation? That'll get you probably full points every time. Yeah.
Could you explain the positive feedback?
Yeah. Which part? Both of them?
Yeah.
Okay, so we1 puts on an inhibitory phosphate on CDK cyclin, and CDC25 goes back and takes that off. But at the end of this process, when MCDK cyclin is activated, it's a positive feedback regulation for CDC25. So it activates CDC25, which goes on and takes off phosphates on other CDK cyclin complexes.
Okay.
So the more MCDKs that get active, the more CDC25s get active, and so for future MCDKs, there's already a bunch of CDC25s that can take off the we1 phosphates.
Okay.
Basically, MCDK is upregulating its own activity, right? Because it's preventing its own inhibition and also increasing its own activation. And both of those are examples of positive feedback.
Okay. Thank you.
Anything else? Cool. Yep, just to recap. All right, and now for M phase. Again, the big marker is the activation of M-cyclin CDK. That was a quiz question, I believe. Why it's important is because it phosphorylates proteins in M phase that are required for cell division. Lamins, condensin, and then I forget what MAP stands for. Microtubule assembly proteins, I think. and other motors. So just generally know that it's an activator for a bunch of proteins. Here are the proteins. And then the big thing that happens in M phase that we talked about was a spindle assembly checkpoint and just generally spindle assembly. That happens during prophase, where the cell disassembles the entire existing microtubule network because it needs to recreate one for 2 cells that'll eventually split. So, yeah, it reassembles all the tubulin that was existing into the 3 types of microtubules that you have: the aster, kinetochore, and interpolar. I think there's a diagram. Yeah. So once those are all done, you start anaphase. I guess, wait, we didn't really talk about metaphase, but
Well, yeah. Sorry, that was the one thing I had in the speaker notes.
that's fine. Yeah. Basically, yeah. Between this happening, you go into metaphase, which is everything lining up in the middle, as a result of all these microtubules, and then in anaphase, it's getting pulled away, split into 2 parts: anaphase A and anaphase B. The chromosomes are moving towards the pole, so away. And the kinetochore MTs are shortening, so that's these MTs that get shortened. And then anaphase B is the push pulling that happens. So the spindle pull. Yeah. So this push pulling that happens in the middle, that's anaphase B. If that makes sense. Cool. Oh yeah, here's a bigger picture. So, you guys understand what push-pull means at the middle? So can somebody explain to me how the pushing works and how the pulling works? You can kind of like use your hands to draw, sort of, but yeah.
Isn't it kind of like they're walking on it, and when they walk on it, they like pull the microtubule towards it, forcing it out more?
Yeah, so they walk against each other, right? So that's the push-pull dynamic. Yeah.
Yeah. There's like 3 different mechanisms of this during anaphase. So you have your dyneins that sit on aster microtubules that are attached to like the cell cortex, and these ones are kind of anchored in the cell membrane. And when they walk towards the minus end, they're like pulling the microtubule underneath their feet, right? Because they can't move, but the microtubule moves instead. It's hard to visualize, but like the way I've kind of tried to think about it is like if you're walking on a treadmill that's not moving and you like anchor yourself in place, you can still push the belt beneath your feet with your feet, right? So that's kind of what the motor protein's doing. The motor protein is anchored in the cell membrane, like you're holding onto the sides of the treadmill, but you're pushing that belt or the microtubule underneath your feet to pull the spindle pull towards the sides. Kind of the same with the middle. So your egg5 proteins are walking towards the plus ends. So as they're kind of attached like this, so as this one goes towards the plus end, its feet are pushing
The other one. Yeah.
What?
Yeah. Sorry, you got it.
Oh, okay. Yeah, and then you have other dyneins that sit on your interpolar microtubules too, that again are anchored here. They walk towards the minus end, and they kind of push the microtubule as these ones are here. They can push the microtubule this way as this kind of walks. Yeah, it's kind of a mess to visualize. I wouldn't stress too much about this for the exam. You might have like one question on it, maybe. I feel like she doesn't usually try to target this stuff just because it's very hard to visualize, right? But yeah, yeah.
So minus end directed motors pull and plus end directed motors push.
Kind of. Here, there's like a. So the minus end directed ones attached to the cortex pull, like they're pulling this apart. And when the plus end directed ones are sitting here, they're pushing these apart. And these like dyneins that are attached in the middle are kind of like regulating this so these don't walk off the end, right? So it's kind of like these are pushing in this direction while these pull in this direction to kind of regulate that. Does that help at all?
I mean, like, how would you
Big-
ask a question if like there were no minus and directed motors?
Right,
And
so-
if they were positive directed motor, what's happening? Is it elongating? Is it shortening?
Yeah. So again, I think that's also why she doesn't really deal with this stuff, is because these are really hard to ask questions about. We have one example of a question she might ask about this after we finish the slide. But I think that's about all you'll get on that. So yeah. And then here's just like a written explanation on that, if that helps you think about it more. So the dyneins in the cell cortex pull the microtubules toward it, providing an outward pulling force on the spindle poles and chromosomes. Your EG5 plus N directed motors bind interpolar microtubules in the middle. Remember, they have like feet above and below their like body. They provide an outward pushing force, pushing those spindle poles further apart. And then dynein, another, again, minus N directed, sits on those interpolar microtubules as well, kind of pushing the spindle poles closer together by pulling those interpolar microtubules towards itself. And this just kind of balances that outward pushing force directed by EG5. And then during all of this too, you have your kinetic cores like depolarizing, and those are also moving towards opposite sides of the cell. So yeah.
Does that sort of make sense for people? Cool. All right, so here's your.
I just left the answer up on this one because like this is, yeah, this is an example of something she might ask on microtubule like push-pull dynamics. So if you have EG5 inhibition, right, so that's pushing the microtubules apart, that's providing the outward pushing force. If you inhibit EG5 or give a loss of function in EG5, then those inward pushing forces provided by dynein that are also on the interpolar microtubules could overpower that and just like collapse the spindle basically. So if the opposite thing happened, like you had a loss of function in dynein or kinesin 14 inhibited on those interpolar microtubules, then EG5's outward pushing forces might cause those spindle poles to be further apart than usual, right? Because you don't have that balance of the dynein pushing in while EG5 pushes out, basically. Yeah.
The dynein thing is like, is talking about like, is an inward and is have outward pull, right? Because like.
Yeah, so dyneins in the cortex have like an outward pull, but dyneins on the interpolars have an inward push.
Should we talk about that in this question about like explaining the dynein into like inward and outward of that?
I think you could.
Yeah. Dynein because like both like or minus and directed.
Yeah, I think you could, and it would again kind of, yeah, it would. I think this question is focusing more on interpolars, but if you were to talk about dynein in the cell cortex, I don't think you would be wrong, right? So yeah. Yeah.
Yeah.
So if you have like in general a so like when dyneins are anchored, rather than moving themselves, they're gonna move the spindle in like the opposite direction. Like if you had one that normally walked toward the plus but it's anchored, then it would move the spindle in the minus direction. Or
Yeah, just think about it like moving it in the
Yeah.
opposite direction of the way it's walking,
Yeah.
right? So like when you're walking,
Yeah.
you're pushing down on the floor, right?
Yeah.
And so it's pushing things back
Yeah.
or pulling it back, but basically, yeah. Okay.
All right, so this is the big thing, spindle assembly checkpoint. This is how the cell will inhibit like segregation until the proper kinetochore microtubule attachments are made. Because again, you don't want to pass through the spindle assembly checkpoint prematurely. Does anybody remember what happens if you pass through prematurely? Yeah.
Mechanic chromosomes get pulled apart incorrectly.
Yep.
Or like not pulled apart.
Yep, and so you might have extra copies on one side, less copies on another, which is obviously a problem. So when the kinetochore microtubules, the ones here, from each spindle, they attach to each separate chromatid, and yeah, that's where they attach. They're still held together by cohesin at this point, so they can't actually split yet. Yeah, I think that's pretty much all you really need to know about this checkpoint. I think I'll get into more next slide. Yeah. So this is like the big mechanism of how this checkpoint works, and it's all about releasing the enzymes that hold together the chromatids. So there are two proteins, MAD and BUB, that sense the tension. Once those become inactive, they release CDC20. Once that's released, it can bind APCC, and then that can ubiquitinolate securin, which will activate separase. The way I remember it is, like, once all this happens, separase is active, separase to separate them. So this is a proteolytic enzyme that'll cleave that cohesin protein, or it just cleaves cohesion. I don't think you have to know what it cleaves specifically, but once that gets cleaved, then the sister chromatids can be pulled apart. So APC is involved in targeting the M-cyclin complex for ubiquitinolation. So these levels will drop again in early anaphase because as the cell prepares to finish off the cell cycle, it'll go back into G1 at some point. And at that point, you don't want CDK/cyclin to be high. You want it to be low so the pre-RCs can be made. So those levels will drop at some point in anaphase. And then the nuclear envelope reforms, the spindle unassembles, and the chromosomes decondense once they're kind of separated. Is that cool with everybody? Sick. All right. Based off the spindle assembly checkpoint, what would a loss of function in CDC20 do? And then a gain of function in securin. Yeah.
Well, I had a question kind of like further back, but I also have an answer for this, I guess.
I can answer your question if you want.
Okay, so can you elaborate a little bit on what active CDK cyclin does to help form the pre-initiation complexes?
Oh, like in the early cell cycle in G1.
Yeah.
It needs to disassemble the
What's the
active
question?
CDK to assemble pre-ICs in your topic 7.
Oh, basically, like, she doesn't really talk about the mechanisms exactly. I don't think that much, but the idea is just CDK phosphorylates proteins that will assemble, like form the pre-IC. So like the replosome is phosphorylated and like recruited to that or to the pre-RC. The pre-IC kind of it's like recruited to the pre-RC, and then the pre-IC can recruit like your DNA polymerase, your replosome, stuff like that.
The entire point of the pre-RC is to get helicase there, and then once that's there, then CDK can be activated, bring in the extra proteins that helicase needs to work. That's about it. You need to know, yeah.
Okay, so
Yeah.
helicase and everything before it is the pre-RC.
Yeah.
And
Yep.
then, like, CDK becomes active and then
Some of those proteins go away too.
Yeah,
Yeah.
CDT1 brings
Yeah.
it. Yeah.
Yeah.
But
all
yeah,
the-
helicase is part of the pre-RC. Everything else is pre-IC and beyond. Yeah, and the pre-IC helps recruit the replosome. I think how it.
Yeah. I think that's all right. So did you want to answer this one now?
Okay,
Yeah.
So CDC20 is what makes it, like, activates the pathway to cleave cohesin, so if it couldn't function, then cohesin wouldn't.
Couldn't separate. Yeah.
So chromosomes wouldn't separate.
Yep. So yeah, loss of function in CDC20, chromosomes can't separate. Can't separate means can't pass through M phase, probably get stuck. If this is an unresolvable problem, I think apoptosis is probably what happens, yeah.
I just have a question. I understand that answer, but could a loss of function in CDC20 also be that like it can no longer like MAD and BUB can no longer like affect it, and then since it can't be inactivated by MAD and BUB activity, then would it always bind at APC and it would just go through?
You want to explain that one more time? Sorry.
So
Well,
how,
you said MAD and BUB inactivate CDC.
or like
They release CDC20.
Yeah.
well, like
Right. Yeah.
And does it do that by binding to CDC20 and then like moving it somewhere? So
Oh,
when
yeah.
it lost
Yeah.
a function in CDC20, has it lose the ability to bind to
Oh, I see what you
that
mean.
above?
Okay. Okay.
And now it's no longer being sequestered. Could it bind to APCC and it would just like go uncontrollably?
I think you
Yeah.
could say that it does get sequestered, or that can happen. Then just be clear that like the mutation doesn't affect its
Yeah.
binding site to APCC. Yeah.
Yeah. But the idea here is that it would be like the loss of function is affecting its binding to APCC.
Yeah.
Yeah. But yeah, I guess that's something we should specify. In the
I guess
question.
yeah. I think yeah. We should specify that. Also, I think a way to think about it is like a loss of function is typically about its like primary function.
Yeah.
It's like latent function is to be released from MAD and BUB. But yeah, I think you have a good explanation. Yeah.
Yeah. And also, if Laura does ask questions like this, she'll probably say a loss of function so that CDC20 cannot bind to APCC, and so it'll be more specific.
Okay. What about gain of function in securin?
So, security is what happens like
Yep. Yep.
Yep.
Yep. That's good. Those are both good answers. So yeah.
Okay. Let's take like a 5-minute break since we've still got an hour to go. Also, there's like food and snacks and stuff down the hall in Moral 337 if anyone wants to run down there and do that while we break. So yeah.
Hey John.
Yeah.
I brought my exam tool with me.
Okay.
Because I have a couple of questions, and can we look over them after the?
Yeah, of course.
Would that be okay?
Yeah, yeah,
Alright,
sure.
thank you.
What's up?
What's up?
What's up? Yeah.
Yeah.
I didn't know you guys were musicians like when you said like I think it was a message like I was like, oh, you said you're starting right now.
Oh. Yeah. Show's leaving like right after this, so we need to do it in the afternoon.
Oh, he's on home.
Yeah. Yeah.
This one.
Yeah.
I don't know why I'm confused about it. So,
I mean, it's a mess. Everyone's confused about it. It still confuses me, so.
so dining says dining here and here, and
Yep.
then the EG25. Those are the two head ones.
Yes.
And then is that dining 5? Is it dining 5?
Sorry?
What family is this? 5?
Oh,
Something?
I think so. Yeah, I don't
Yeah.
actually remember that.
But and then those two, they're kind of like they're pulling this one here. This one's pulling this one here.
Yeah, so these are like pushing apart, right? So if you have your egg 5, right?
Yeah.
And it's walking towards the plus end. So that will kind of, because it's technically anchored by its other half,
Yeah.
this microtubule will be kind of moving underneath its feet and moving towards the spindle pole.
It's walking towards this side. So
Yeah.
then it's walking towards this side. Doesn't this make this looser? If it's
What do you mean
walking-
looser?
Like it's walking through this side, putting more rope or like microtube, more rope like here. So then wouldn't this side be looser? There's more.
I don't fully get the rope analogy. I know that's kind of how Laura describes it. How I look at this is that like its feet are like here, right?
Mm-hmm.
And so the microtubule's here, and as it walks, like it's pushing the microtubule backwards.
Mm-hmm. So this one right here.
Yeah,
It's
it's doing the
pushing-
same thing, but in the opposite direction, right? Because the plus end is over here. So it's walking in this direction and pushing my arm.
That way. So
That
it's
way.
pushing it this way.
Yeah.
Oh, both of these are pushing them
Yeah,
this way.
they're
This
pushing
one's-
in opposite directions. So your egg 5's at the center.
Okay, wait. It's pushing this one this way, that one that way.
Yeah, exactly.
That. So then so the only thing that's pushing them closer is this.
Yeah, it's
Dining.
it's this dining that pushes it closer. right? So it's walking towards the minus end.
Yeah,
So it's
it's
pushing it this way. So because it's anchored here, right? So it'll push
Mhm.
this one this way. But this one that's anchored at the spindle pole is going to do the same thing, right? Because it's also walking towards the minus end and pushing it this way. So these dyneins that are like on the interpolar
Yeah.
microtubules are stabilizing that action from egg 5.
Mhm.
So they're pushing them towards the middle.
And this is just keeping all this still. So
Yeah.
this is pretty much still.
For the
Most- or like
for the
stable,
most
or like
stable,
this
yeah,
part.
yeah. Stable is a good way to say
And
that.
then-
Because ultimately you need these at the ends of the cell instead. But if you didn't have a force kind of keeping it in check, then
Yeah.
you just push it too far, or these motor proteins could like walk off the microtubule or stuff like that.
Yeah.
Yeah.
So this is just holding it on, like holding, what's these called?
Those are spindle poles.
Spindles. The spindle poles. They're holding the spindle poles in place. That's the whole purpose of this part. So then microtubules can grow and see if they can attach to the kinetochore of the chromosome.
Yeah, and then like when you have the push and pull apart, like these shrink, right? And
Yeah.
these pull the spindle poles. The spindle poles still need to migrate to the opposite sides of the cell.
Yeah.
And these do help with that. But if you had like
These just make sure that the chromosomes it doesn't pull it, like it prevents these from pulling all the way back.
it prevents it from pulling it too far, yeah.
Okay.
But these are still like mechanisms of separation. So if you go back to the this one, right?
Oh,
so
could you explain like how they would what they would be doing each thing right here?
yeah, so anaphase A is when you're pulling your chromosomes towards the pole itself.
Yeah, and then these are the what's it called, EG25?
Yeah, those are your interpolars. So interpolars are not involved in anaphase A. So anaphase A is your kinetochore microtubules, right? And so it's depolarizing at this like kinetochore,
Yeah.
and then separating those chromosomes,
Yeah.
and then pulling them towards the pole itself. Anaphase B is that.
Wait, this is just through depolymerization and polymerization. There's no motor proteins involved in this,
There is a motor protein that causes that depolymerization at the kinetochore.
but not these.
No.
Okay, so there's another motor protein bringing it down, but this whole thing is caused by.
Yeah, so the idea of anaphase A is you're moving the chromosomes themselves towards the spindle poles. The spindle poles are locked.
Yeah.
Essentially. The idea of anaphase B is that now you're moving your spindle poles with those shortened chromosomes.
Oh, because they can only go so far. Like
Yeah.
they're not reached to the each side
Mhm.
yet. So then you have for this one it extends this part so they can fully reach this
Yeah.
other side.
Yeah.
I know.
Okay.
Yeah. Okay. So think about this, right? So
Maybe
this
I'll-
is the dynein anchor.
Thank you.
Yeah.
That's it.
And those kind of like pull, right? They
Like
pull.
the treadmill thing. Like the
Yeah,
treadmill
yeah.
analogy.
Keller had the treadmill analogy. I kind of like to think of it as like tug of war. Like you're pulled. These are like with their feet, kind of pulling these aster microtubules. They're pulling that pull this way, right? Versus you have the dyneins in the middle. These are the interpolar microtubules, right? Think about it like you're on two ropes, right? You're holding the top rope, and you're like pushing the rope backwards.
So
Right,
you're staying like
so you're,
stationary.
for the conception, you're staying stationary.
Okay.
The pole is that way. You're like dragging the rope backwards towards that pole,
Okay.
and you're holding onto the top one.
And then, wait, what's the one that's going backwards? Like this bottom part is what's going backwards?
Yeah. I mean, it
So-
happens both ways, but yeah, you can think about it
Oh.
that way. Well, like if you think about it, you're holding one, you're pushing the other one, they're getting separated. So one's kind of staying still. It's actually also like moving. It's like conceptually kind of dense.
Okay.
Yeah. Wait, why do we need them? Because that's how that's how anaphase happens. Like,
Oh, anaphase B.
yeah. Sorry, I didn't have a good explanation up on stage.
It's like the only reason I was able to explain that is because I happened to do the topic review. Otherwise, I would have never been able
Dude,
to.
this was like last year for 151. There's one topic. I think it was topic 10. Like,
Ooh.
did you come to that one?
No, I was doing it with you.
Yeah, you were there. Oh yeah, you were there with me. Yeah.
What's it called? I know exactly what you're talking about. It's about.
I think it was Anaphase.
Yeah, and then in the final review, I had to do it again.
Mhm.
That was the only way. It was meiosis.
I was meiosis.
And you know how there's so many confusing words in meiosis?
Bro, I don't remember meiosis at all. Oh my god.
That took me so long just to get, and there's only one person who showed it to our session. So like that was good because I was able to just sit down and explain it to them.
Yeah.
This one?
Yeah, there you go.
Is everybody?
Okay,
Or is
so
it backwards with the arrows?
well, no, it's context dependent, right? So like, if you're saying he's walking this way, then I would say that the pole is over here,
Is everybody to the left back? I think we're missing one person.
right? And so this one's getting close. This one's like semi staying still, semi going that way.
Wait, if he's walking this
So
way?
you're.
I thought you said he's
So
like
you're yeah.
kicking it
He's
backwards.
kicking it backwards, right?
So
Towards
what
the
if you're
pole.
walking the other way?
Yeah, technically.
But it's not.
The pole is moving in the opposite direction of the way it's walking.
Yeah.
Okay,
That's a
so
good
the
way.
pole's the kicking backwards. Because the pole is moving the opposite way you're walking.
Yeah. So like.
If you think about it, like,
How do you recommend cramming?
I wouldn't.
Well, it's like
I don't think you need to cram, yeah.
but it's like, it's like too slow.
What, is studying?
Yeah.
What are you on?
Well, I like went over all the homework, but it's like that's what I used to practice
Yeah.
myself. 'Cause I'm too tired of us searching anything else anymore. It's just homework and quizzes. But it's like it takes me forever to get through them,
Mm-hmm.
and I feel like I'm not retaining as much information as I would like to.
Okay.
I'm at the point where I'm like I think I should just focus on things I don't know at all, or I don't know with confidence.
Did you go to the topic 1 review?
Yep.
Okay.
It was mainly this stuff. So
Oh,
9
okay,
to 10,
okay, okay.
Where I was like
Yeah.
telomerase.
Yeah. I mean, we'll get to that. But if you have questions at the end, you know, I'll help out. And then, I don't know.
I just feel like my way of studying is like it's not like flashcards. where it's
Yeah.
quick.
But again, flashcards isn't really the way to study for this class anyways.
Yeah, I wish you took like last year's exam and gave it as a practice exam.
That would be nice, yeah.
She's probably gonna recycle some of those questions.
Yeah. I mean, that's also why I try to work some of these questions into here. Like, I took some questions just directly from last year's exam and put them in the review session. So, yeah, I don't know. I mean, I say this to everybody, but it's just like covering, and you kind of do this too, like covering answers and then trying to answer the problem on your own from like homeworks or quizzes or whatever is always helpful. Just like, I don't know, like writing stuff out is always helpful. Like I used to flip through my notes and just like rewrite some stuff.
Yeah, I ran out of so my history notebook. It's now sharing space
Nice.
with biology.
Yeah.
I do. I like the wording.
But yeah, it would be helpful if she included like a exam review sheet or like stuff you need to know or just like practice problems or something. Yeah, I'll bring that up to her for future use. Unfortunately, I can't help you now because it's.
This is more like tonight, and I'm gonna go over everything I don't know.
Yeah, I think you know more than you think you do because you always say you don't know stuff and then you always like kill the quizzes. So, you'll be fine, I think.
The quizzes are more like homework. The exams are like random.
I think the quizzes are pretty akin to the exams. I don't know. I mean, the exams just have more yapping, in my opinion.
Yeah, a lot.
There's a lot of yapping in the problems, but then the actual like question is not that like in
5-page
depth.
essay, and then it's like, okay,
Yeah.
so what happens in a philosophy? Like, really?
Yeah, yeah.
She has to abridge somewhat just from the amount of content she's covering, but
Yeah.
maybe.
Typically, the final exam, open responses are like a little shorter. I think you'll have 2 to 3 of them that are more like matching type stuff, and then they'll,
I love matching.
yeah. Yay. Okay. Yeah.
I started studying yesterday because I got done with the other one.
All right. I think that's about good. I think pretty much everyone's back. So we will start with everybody's favorite DNA replication, because who doesn't love directionality? Okay. Yeah. So just a quick overview of DNA. DNA, as we all know, 2 complementary strands that base pair to each other. they have a 5' end and a 3' end, and these kind of are anti-parallel to each other. So you have 5' end at one end, and your 3' end, like matching that side, and then swapped. Yeah, I don't know how to really say that. Just 5 here, 3 here, 5 here, 3 here. DNA is made up of a phosphate, a sugar, and a nitrogenous base. Your phosphate group is at the 5' carbon of the sugar, and DNA is also composed of, it has one hydroxyl group at the 3' sugar. Yeah, your nitrogenous base is attached to the 1' sugar, and your nitrogenous bases hydrogen bond with each other when forming a complementary strand. So adenine and thymine form 2 hydrogen bonds, while cytosine and guanine form 3. And then, of course, you also have uracil, which is the RNA nitrogenous base, and that binds with adenine, also forming 2 hydrogen bonds. The bond linking the backbone, it's like the sugar phosphate backbone, is called a phosphodiester bond. And so those link your nucleotides in a strand. Yeah, I think that's about it. I don't know, I feel like we've been working with DNA at this point for a couple semesters, so should be okay with that. Yeah, so DNA is always synthesized from 5' to 3', and that's because of this nucleophilic attack that happens. We don't really need to know like the super specific chemistry details of this for this class. In my opinion, it's just helpful to like visualize what's actually going on and help you remember like the directions of synthesis better. So you have your oxygen on your 3' carbon, which performs a nucleophilic attack on the phosphate of an incoming nucleotide, which is located on the 5' carbon. The energy for forming this bond comes from cleaving off these 2 phosphates, which is called a pyrophosphate. And this is the reason basically that you always need to add to a 3' end, because you need these 3 phosphates here to be able to have the energy available to create a bond between 2 nucleotides. Yeah, again, the bonds between the backbone, the sugar phosphate backbone, are always covalent bonds, while the bonds between nitrogenous bases are non-covalent bonds or hydrogen bonds. Yeah. So replication, this is called the semi-conservative model of DNA replication. It basically just means that when the parent strand is replicated, one strand goes to one new strand of DNA, while the other strand goes to the other. And so you end up having this parent template that stays through all the way to here, and then this lighter purple one becomes the parent strand just to form that one. Yeah. Okay. So cells start replicating their DNA at the replication origin in the S phase. This is what we just talked about. So you have ORC, which is where that origin lysing happens, and allows for the start of DNA replication. So cells have multiple origins of replication because we have billions of base pairs, and so it would be extremely inefficient if we only had one and had to build out from that. Which means these replication bubbles can join and meet and form larger bubbles, basically. Replication occurs bidirectionally, so there's 2 replication forks. You have your origin of replication at the center, and then you have a fork moving in this direction and a fork moving in this direction. Yeah. At the fork, you have a bunch of proteins that are all involved in synthesizing new DNA strands. So you have your DNA polymerase, which of course is actually adding the nucleotides and creating that strand. You have RNA primase, which lays down a short RNA primer that the DNA polymerase uses to synthesize from. You have sliding clamp, which kind of keeps DNA polymerase adhered to the strand of DNA. You have single-stranded DNA binding proteins, which as helicase comes along and unwinds the DNA, single-strand DNA binding proteins can bind to that DNA and basically protect it from the cellular environment because single-strand DNA is not super stable. What else? That, I think, is everything important that we talk about in this class. Yeah. Yeah, so helicase, DNA polymerase, sliding clamp, primase, and single-strand DNA binding protein. And then there's also, sliding clamp loader, which is an ATPase that uses energy to put the sliding clamp onto the DNA. Yeah. And so kind of building upon what I talked about earlier, the idea of the primers being added onto DNA. When you have a single strand of DNA, your DNA polymerase can't come and just synthesize DNA out of nothing, right? It needs a 3' oxygen to be able to start adding nucleotides onto. And so primase lays down a short, 8 to 10 nucleotide RNA primer that provides that 3' end to begin synthesizing your DNA off of. Primers are removed after synthesis and filled in by DNA polymerase again. So once this happens, we'll take this primer off and then synthesize over it with a new DNA polymerase. The reason we use RNA isn't totally known, but the thought is that it's basically forms a less stable kind of connection or hybridization with DNA, and so that it's easier to remove at the end of DNA synthesis to replace with DNA. Yeah?
Do we know why the RNA is less stable? Because I know that normally RNA is single-stranded, but in this case it's not.
Yeah. It just...
It's the bind with uracil or something like that.
Yeah, it just... DNA bonds are strong, RNA bonds are strong, DNA-RNA less so, right? Annealing to each other is not as complementary or stable, I would say. It's still stable, but it's not as stable as a DNA-DNA bond would be. Yeah, and so now we have to move on to direction of synthesis. So yeah.
There's definitely, almost definitely going to be.
You will have questions about this. Probably pretty similar to your first question on quiz 9, where it gave you the whole replication fork and you had to identify different parts of direction of synthesis stuff, label the strands, stuff like that. So yeah, I think the key thing to always think about, right, is DNA is always synthesized in the 5' to 30' direction, no matter what, right? And so if you have a template strand that is on this side, 3' to 5', DNA polymerase will be able to read this 3' to 5' and synthesize 5' to 3'. And because it's able to synthesize 5' to 3', this one's your leading strand because it's able to continuously synthesize that strand, right? Because it's just going in the direction it needs to. It's going 5' to 3'. The issue becomes when you come to the lagging strand. And so your DNA polymerase is on the template strand, but it's reading at 5' to 3'. And if you're reading something 5' to 3', you can't synthesize something 5' to 3', right? Because it has to be anti-parallel. It has to be complementary to that. So this has to be 3' to 5'. So, but because DNA polymerase can only synthesize in the 5' to 3' direction, this is why we get the problem of Okazaki fragments. And so each Okazaki fragment requires a primer, and because your polymerase can only synthesize 5' to 3', you'd have a primer added here and synthesize. And then your DNA polymerase comes off, primer here, synthesize. Primer here, synthesize. All while the direction of fork is moving this way. So if you are synthesizing in the opposite direction of the fork, right, you're building your DNA out this way, then you are building the lagging strand. But if you're following the direction of the replication fork, so everything's moving this way, and you're fine, you're just moving your DNA polymerase along, then you are synthesizing your leading strand. Tips for this: always label your ends before you start any of these problems. Make sure you do that. Think about the fact that you need to add a new nucleotide to the free 3' end, so you don't mix yourself up. DNA polymerase reads 3 to 5, and it always synthesizes 5 to 3. Yeah. Here's a little bit better of a diagram of just the lagging strand zoomed in. So this is now moving in the opposite direction. If we have our fork going this way, so down here, our leading strand's going along because it's reading 3 to 5 and synthesizing 5 to 3. Here, our DNA polymerase can't do that. So we lay down a primer and we synthesize 5 to 3. And then we move down, we lay down a primer and we synthesize 5 to 3. primase puts a primer here. DNA polymerase jumps over here, synthesizes 5 to 3. And so this is like the ribosomes moves at the same time, right? So this is kind of connected to your your DNA polymerase that's synthesizing the leading strand. So these are moving together down the fork, but this has to like kind of do this while that's happening. It's also kind of annoying to conceptualize, but that's how it works. Numbering your Okazaki fragments. So if the fork is moving in this direction, your fragments farthest away from like where the fork is are going to be were synthesized first. So this would have been synthesized first, then second, then third, and fourth, and so on. I think that's all I have to say about that. And then once the DNA is all synthesized, your primers are removed by an RNase, and DNA polymerase goes back in to fill these spots with DNA nucleotides. So once this is kicked off, we have a free 3' end here for DNA polymerase to basically just synthesize up until this nucleotide, at which point DNA ligase can come and seal this final bond between those two DNAs. Yeah. Any questions on direction of replication, lagging strand, leading strand stuff? Yeah.
For that like, original diagram you showed,
Yes.
I don't know if this is a confusing question, but if you switched all the directions of the arrows, could the leading and lagging strands switch? Like, what I'm saying is like it doesn't matter if it's the top or the bottom.
Yeah.
It's just
Yeah.
like,
Exactly.
Whether or not like.
So you're saying if your 3' was up here, right?
Yeah.
And your 5' was here instead?
Yeah, like it's just all relative to.
Yeah,
Yeah,
we're
it's all relative to that. Yeah.
Yeah.
As long as it's opposite.
Yeah.
Okay.
Yeah. It's all, I wouldn't get stuck up on top or bottom. Just look at what strands are actually there, 5', 3', and then figure it out from there.
You might have a question about which direction the fork is moving, left or right, and that's all, again, relative to setting it up properly.
Yeah. Yeah. Okay. Just like a quick question about this. This was on your problem set recently. So this gives you 2 strands. It doesn't really show you the replication fork, which makes it a little trickier. But yeah, so one, replication of this strand involves several primer molecules. Is this strand A or is this strand B? Yes. Yeah, it is strand A. Oops, sorry. Because again, strand A is 5' 3', and this is the template strand. So if DNA polymerase is on the template strand and it's moving down the template strand, it's going to be reading this 5' to 3', which it can't do, right? So that means it has to be reading at 3' to 5', which means you're synthesizing Okazaki fragments. And since Okazaki fragments require multiple primers, that strand A, is going to require several primers. I've already revealed the answer for 2 accidentally, but yeah, the DNA polymerase will always move 3' to 5' on the template strand, no matter what it is, no matter what strand it is. Even though the whole fork is moving to the right, DNA polymerase is still moving this way on the lagging strand, and it's 3' to 5' on the lagging strand, reading in that direction, synthesizing in this direction. And then again, the leading strand DNA polymerase is reading 3' to 5', and synthesizing 5' to 3'.
Wait, but it says it moves 3' to 5'.
Yeah,
Those moves mean like reads.
basically, yeah.
Oh, I thought it meant like synthesized. Oh.
Like the whole fork is moving,
Yeah.
but the actual DNA polymerase, right? It's still moving on that template strand, 3' to 5'. Because if you're doing your Okazaki fragments here, then yeah, okay, cool. Yeah.
Okay, so I think is the reason that the answer is A because of where the origin is,
Yeah.
and because A is 5 to 3 away from the origin.
Yes, exactly. So if the origin's here, and we move outwards from both sides of the origin, right? This is your 5' end, this is your 3' end, and so again, you have to synthesize 5 to 3. So it means you have to be synthesizing this way on the origin. So you have to be doing this.
Okay, and then so would that origin be like, is that where like the bubble is? Like,
Yeah, this
Yeah.
origin is where this bubble is opening, and then
it's
starting
going
out from this
this way overall.
Yeah.
Okay.
Yeah.
So we're supposed to think of the
strand that's being made, not the 5 tree we see there.
Correct. Yeah.
The one that's being made.
Because here, I think in the problem, she said here are 2 template strands, which I didn't specify on this slide, but yeah, these are your template strands, and then you're synthesizing off of these. Okay. Now we get to the end replication problem. So this occurs on the lagging strand when the primer from the last Okazaki fragment gets removed. And this happens because there's no 3' end to add DNA onto, right? Because you can't break a pyrophosphate off and get the energy to cleave that if you're not adding onto a 3' end. So this end of replication problem is basically just every time DNA replication occurs, we're losing this small little section at the end of the lagging strand because this primer is removed, but there's nowhere for DNA polymerase to add more DNA onto it. This would lead to ends of linear chromosomes shortening over time. Yeah. And then this is, of course, fixed by telomerase, which I will ask a few questions about first. So what type of enzyme is telomerase? I've had this question like twice now, I think. Yeah. Exactly. It is an RNA template-dependent DNA polymerase because it has a built-in RNA template, but then it synthesizes DNA. It actually makes DNA. And then what strand does telomerase extend? Sorry. Yeah, the lagging strand template strand is what it actually extends. So the problem occurs with the lagging strand, but telomerase isn't actually doing the synthesis of like filling in that primer. It's extending the lagging strand template strand, which I will talk about now. Here's your answers. So yeah, telomerase is an enzyme that carries its own RNA sequence. It binds to the lagging strand template strand here and is able to basically build this out. So right, your lagging strand template strand is still 5' to 3', and telomerase is a DNA polymerase, so it still has to build 5' to 3'. So by binding to this lagging strand template strand with its RNA template, it can basically extend out this lagging strand template strand. Only then can now an RNA primase come in and add a new primer onto this extended strand, and then DNA polymerase can come back in and move 5' to 3' to fill where that primer was. So we're not, I think a common misconception with telomerase is that once that primer is removed, telomerase just comes back in and fills it in. That's not the case, right? Telomerase is extending the template strand so that another primer can be added, so that DNA polymerase can come back and add off of that second primer, basically. Yeah. The telomerase template has 2 nucleotides that actually bind to the end of the template strand, and then it has a repeated sequence that is used to extend the template strand. And I think that's pretty much all you need to know about that. Any questions on telomerase? No. Okay. Here's just another diagram. So you have your 2 nucleotides binding to the lagging strand template strand. It extends this strand out. A primer comes and adds here, which is not pictured here, but then DNA polymerase can add from this primer, get to the spot where ligase comes in and fills in that gap. Yeah. That's all I have for topic 9. Any questions on that? DNA replication, directionality, telomerase. Yeah.
What are the odds if she asks any questions about the first slide that you guys have, with like chemistry and stuff?
Oh, very low.
Okay.
Yeah. Just know that it adds to a free 3' OH, but the nucleophilic attack stuff we don't care about. I just find it helpful to include because I think it's easier to visualize why it's actually happening, but you don't need to know that. Yeah.
So here for this, is the primer being added to the lagging strand after the telomerase extends the template?
Yes. Yeah, it's part of the lagging strand, right? It's being added complementary to the lagging strand template strand. And so that primer is then built off of with DNA polymerase, but then removed later again.
Okay.
Yeah, does that make sense? Yeah.
So telomerase is what stops this problem from happening. But what does the telomeres do?
The telomeres?
At the end.
Oh yeah. So not all organisms have telomerase. Humans don't actually have telomerase, which is right. I don't think we do. I think only certain like cell types have telomerase. So
Yeah, I think sperm cells have telomerase. But
yeah, yes, they do.
I think I'm talking about the caps at the end of the chromosome.
The telomeres.
Yeah,
Yeah.
yeah. So these are the telomeres. It's extending the telomeres. What are you asking about?
No, because this confuses me. Because if we had telomerases, then wouldn't we would have run out of telomeres, no?
Correct. Yeah. But we don't have telomerase, which is why. So like yeah, it's we have telomeres so that we don't actually like get rid of actual genes that we need, right? So as our cells divide, these telomeres constantly shorten, but they're so long that it doesn't really matter until we're like super old. Some organisms are able to like maybe their telomeres aren't as long or something. They're able to they have telomerase encoded that can extend their telomeres, so that they basically never lose any DNA in successive replications.
Other cell types also have circular DNA, and so they don't have a use for telomeres.
Yeah.
Yeah.
Isn't it basically that like we have telomerase in like stem cells and sperm and egg cells, so that we produce all the telomeres we need for like an entire life when we're born, and we don't
That
we just go off of that.
could-
and then
Yeah, that
the
could be
next
part of it. Yeah.
generation gets all the telomeres it needs because there's
Yeah.
telomerase in just those cells.
I think that's part of it. Also, from what I've heard, it's like all those germline cells, like sperm cells, as well as some types of stem cells. They have it because they haven't been differentiated yet, and so they need to conserve every single gene that's on the
Yeah.
template. And so that's like one of the requirements of it. But like in normal adult cells, like usually the genes at the ends don't get transcribed anyways.
Yeah.
So they don't really have a need for the caps.
Yeah.
Yeah.
If you're a single cell organism, where like the next generation is a single cell cycle, then you need telomerase the whole time.
Yeah. I would
yeah,
just
like a stem cell. Is that what you're saying?
Or just like a bacteria.
Or like amoeba? Okay.
Yeah, yeah. I mean, like, this isn't going to be important for the exam, right? Like, it's just,
Common knowledge.
yeah. Really, the only thing you need to talk about, like, when she asks about what is the end replication problem, you just say there's DNA lost when the primer is removed from the lagging strand. Yeah. We don't. It is interesting, but, like, she's not going to ask you about, you know, why telomeres and stuff like that. So, yeah. Okay.
All right. Almost done, guys. Hang in there. We got DNA damage and repair. So DNA polymerase has an inherent function DNA has another inherent function of proofreading. So this is different from like repair. This is proofreading as it goes. So the DNA has an exonucleus domain that kind of checks its work, so to speak, which reduces the amount of errors from a lot to a little. I don't remember the exact number, but essentially the way it works is it moves the incorrect nucleotide that was just added into the exonucleus site. The exonucleus has 3 to 5 prime activity to go backwards and cut it off. I guess I just answered my question there, but because DNA synthesis happens 5 to 3, the exonucleus works in reverse, 3 to 5, to go back and correct it. Yeah, like as you can see on this diagram, DNA polymerase is working to extend this top strand. Here's here's the incorrect one that gets added on. It doesn't form a base pair it doesn't form a pair with the other base, and so it's not gonna connect. It's gonna be off like that. The exonucleus function goes back, and then the right one's added in. It's pretty straightforward. So mismatch repair is something that's after DNA replication. So this will go back and take something out and fix it. So Laura might refer to this, like any mismatches you see, as lesions or distortions in DNA, because when a wrong nucleotide is in, it won't be flat, like parallel flat. There's going to be some like bumps and ridges. So that's what the distortion is. And so like humans, any organism that have MMR mechanisms will sense that out, remove the problematic one, and be good to go. So there's a few problems that we talked about if this mismatch doesn't happen or if it happens incorrectly. Can somebody tell me what those problems might be? I guess you can start by telling me what would happen if mismatch repair doesn't happen.
Then the mismatch would proliferate into future generations.
Yeah. Yeah, you'd have unmatching DNA in future generations if there's a mismatch. What about if the newly synthesized strand was not, identified, and mismatch repaired the wrong strand? What would happen then?
If it repaired the template strand, then both strands would be wrong, and then the replicated DNA would be entirely mutated.
Yep. Yeah. So basically, it's important to have the right strand identified. So after that right strand has been identified, the mismatch is identified, the whole area around it's removed, not just that one pair. So there's like an identifying region, and then between that and the mismatch is kind of the chunk that gets taken out. And it works, again, using helicase, separates the template. Exonucleus will remove all the wrong region, or like that whole region that has the wrong nucleotide, and then sliding clamp in DNA, come back, fill it in, and then again it's sealed by ligase. So the addition part at the end, like the right DNA getting added, works the same way. It's just that this is what happens when there is a sense of a mismatch. So here's a question. How do you know which strand is the one to repair? What? Yeah.
The template strand is methylated,
Mm-hmm.
and the new strand isn't, and it can only like fix the new strand that's not methylated.
Yeah. That's one thing, yeah. That happens in E. coli. But in eukaryotes, it's identified because there's a lot of nicks. And so those don't happen on the template strand, those happen on the new strand. And so before ligase comes in and seals everything, that's when the mismatch repair comes in and does its thing. That makes sense. Cool. Okay, so aside from mismatches, you can have DNA damage. There's three main types that we talked about. There's a couple others, but you don't really have to get into it too much. Depurination. So the purine bases are removed, and this becomes like an abasic nucleotide. Abasic just means has the phosphate and sugar. And this is just like a spontaneous damage event that happens. I think it can happen because of UV light, but not completely sure. Deamination changes the cytosine to uracil. And if you look, the structures of cytosine and uracil are pretty similar. There's one terminal amine group on cytosine that gets changed to an oxygen. And so again, this is just a spontaneous reaction as well. Happens, I'm pretty sure, with reactive oxygen species, but you don't really need to know that. But it becomes uracil. And the last one is pyrimidine dimers. So these are also happening by UV light, but two pyrimidines that are adjacent to each other, the nitrogenous bases actually bind to each other instead of kind of staying separate. And so this causes lesions because it can cause the DNA to be distorted in kind of weird ways and fold wrongly. Yeah. So what are the two ways that the cell deals with DNA damage?
Either mismatch repair or basic excision repair?
Yeah, we talked about MMR for like mismatches, but for DNA damage, basic excision's one. Does anybody know the other one? Did you say it? Yeah, NER, nucleotide. Yeah. So just the specifics. Yeah. So for base excision repair, it's in the name. It's just taking out the base, not the whole nucleotide. And this is used for depurinations and deaminations because, again, those are issues with just the two, or it's just an issue with the nitrogenous base. So for deamination, the uracil is identified, and then the bond between the sugar and the uracil is cleaved, so this becomes abasic. And then now that it's kind of like a depurinated one, the endonuclease can come in and cleave the phosphodiester bonds on either side, and then remove that abasic nucleotide, and then DNA polymerase will fill in the missing one, and then get sealed. So pretty simple, straightforward. And then nucleotide excision repair. This happens for that pyrimidine dimer because it's kind of a bigger lesion, causes weird distortions throughout a segment of DNA. So that lesion is identified by a protein. I don't think we have to be too specific there. But that specific protein has endonuclease activity, and it'll cut on either side of the lesion, remove that entire kind of chunk, and then it'll get filled in, same as before. So, given what both BER and NER are, what would happen if you had a loss of function in nucleotide excision repair? So think about like what happens if DNA gets damaged, and then if it can't get fixed. Yeah.
Well, you would still have a pyrimidine dimer there, and since that's such a bulky lesion, it can cause double-strand breaks.
Yeah. Double-strand breaks. So yeah, pretty, again, in the name, it happens when a piece of double-stranded DNA is broken. So UV is a big one, radiation, or unrepaired lesions, like we just talked about. So this can disrupt genes, transcription, kind of the whole 9 yards of things. And then, either cause the cell to die or overproliferate. Those are kind of the main two pathways a cell goes down with DNA damage. So there's 2 ways to repair it. Non-homologous end joining. So this is kind of like identifying both sides, or like both kind of broken points. Those are cut out by exonucleases into like an even chunk, and then they use this non-homologous end joining to just close that gap. Yeah. Do you have a question?
So why is it that they have to shear down the ends before they reconnect it?
Those double-stranded breaks can happen by a dimer or even like loss of nucleotide, and especially with those, a whole frame can be shifted. And so I'm pretty sure it's processed down so that the frame is like maintained. Okay, yeah, so the pros for this method is like you don't need a homologous strand specifically, so like any break can be quickly repaired. But because there's like a lot of processing to even out that chunk, there's proteins or amino acid codons, like the set of 3 that can be lost, which can result in pretty serious changes to like protein folding, but in the overall grand scheme of things, it's better than a complete frame shift, which makes a whole new protein. So the other one is homology-directed repair. So there's a template that comes in and perfectly like repairs it, but obviously it takes more time, takes more resources, and it's usually happening in segments of the gene that like have very crucial information, but again, you need more resources. You need a whole new strand as a template, and it'll take longer time, etc. So yeah. those are those two. I think that's all I have for topic 10. You guys have any questions? Okay, we have like a couple Laura. Oh yeah, what's up?
Do you know how she does the multiple choice questions?
That's what we're about
Yep.
to get to.
Next thing.
Yeah, so Laura's multiple choice questions are not typical. You might have like 10 of them that are normal. Which one is this? A, B, C, or D? But most of them are like this, where she talks, says a bunch of irrelevant information for like a paragraph, and then actually tells you what you need to do, and then gives you like assigns basically a letter to each thing. So in the past, she's done stuff like a massive fill in the blank thing where it's like there's blanks and then you have to say like this is blank number 13, this is blank number 14, and then she gives you like a list A through E of like different proteins, and then you have to like assign them to different blanks in the sentence. Or something like this where she'll give you like a list of things that are happening and then ask you to assign like the phase of the cell cycle that it's in, or I don't know, stuff like that. So yeah, most of her multiple choice questions are like one scenario encompassing like 8 questions at once. So you're gonna have like, I don't know, I think she said 35 multiple choice questions. Yeah, 35. Like so you'll probably actually have 7 scenarios, each with 5 questions that pertain to that scenario, and then you actually have to match that to this. So yeah, if you guys want to take a moment and just practice this, we can reveal the answers after, and then we'll do that again with another question.
Yep.
Yeah.
So would you say it's kind of like similar to when she does like clicker questions and
Yeah.
like you have to like match a number to a word, it's like really complicated?
Yeah, it's more like
multiple
that.
numbers in an answer, that sort of thing. Yeah.
Laura multiple choice is not multiple choice.
They're disguised short answer.
Yeah. They are disguised short answers, yeah.
That's better than like A, B, C, D, all the above, none of the above.
I hate all the above.
Really above.
Yeah.
Or like my favorite, when E is the answer is A. Okay, great, now what?
Yeah.
What's our time?
2:40.
Not bad.
Pretty good time.
Yeah.
What was your question? I'm excited to see your question. Oh, I remember what your question was. Okay. Never mind. Markers. I typoed phosphorylated. Work is phosphorylated.
Phosphorylation.
Phosphorylation. When are you coming back to campus again?
What?
When are you coming back to campus?
Around like
break?
the 5th or 6th.
Okay. Are you just working in lab?
And at Cooley.
Nice.
Jason was babysitting the ER doc heads, or wait, how do I say this? The head ER doc's kids.
That's funny.
Yeah.
Like recently, or?
Yeah, and so
Okay.
like he had just left. Like I said bye to him,
Mhm.
and then he went back to home to his kids, and Jason was there saying bye to him. And then Jason goes, "Oh, by the way, like, here's who I babysit." I was like, "Oh yeah, I know him."
That's funny.
Yeah. Yeah. What's up?
These questions, is it safe to assume that each choice is a single answer?
No, that's
Not
not safe.
always. No, that's not safe.
There will be repeats.
It did happen to work out this way, I think, this time. Oops, did I not put the answers there? Oh, I forgot to. Oh, what?
I probably just have to refresh it.
Okay. I forgot to animate the bar to come off. So if you
Maybe now.
No, I think you just need to delete this bar.
Okay.
Woohoo! Answers. Yeah. So yeah, in this case, the answers happen to be one for each, but that will not be the case.
Yeah.
Always. Yeah. I think a general rule for multiple choice is never read into like how many B's you're putting in a row or how many like of the same things you're assigning. Like it could just be that that's the actual answer.
The professor's not looking into that and making a.
Yeah.
I did too many B's in a row. They're not really.
Yeah. Yeah. Okay.
These answers make sense, everybody? Sorry. Yeah.
Can you explain one? Because I thought that CDK activity was low at the beginning of each phase.
Well, technically not, because if you think about like M phase, right? You jump right into M phase with high and active levels of M cyclin CDK, right? And then, I guess in G2 it's also low, but
No, I wouldn't say so. Yeah.
yeah, I mean like it's not necessarily low. It's really only low in G1.
Yeah. The idea here is that you need low CDK activity to allow for pre-RC formation, right? So.
And then I have a for 3.
Yes.
So wait. Oh, so C would be the answer, but isn't like. So I thought that all that like CDC 25 and We1 and everything was happening like to activate CDK all the time.
Yes, but the CDC25 pathway is specific to the MCDK. So like that We1 and CDC25 like positive feedback loops that are moving from G2 to M are specific to like G2.
Okay.
Yeah.
So for the other phases, CDK is just being activated by cyclin and CDK activating kinase.
We1 is active whenever, so like you could say that We1 is inhibiting in any phase. Just We1 is like the whole point of there are multiple like ways to inhibit CDK throughout all this cycle. So you have CKIs, you have We1, stuff like that. The whole idea with We1 in this G2 transition is just that it's like basically holding a cascade of MCDKs ready to be activated all at once by the stripping of that phosphate by CDC25.
Okay.
Yeah.
All right, we can move on to the next one. Here, just for each scenario, write down whether this situation's happening 3 to 5 or 5 to 3.
Yeah. This would be like an A-B situation.
Yeah, this is like a, 3 to 5 is A, 5 to 3 is B, and then write down A, B, whatever.
Yeah. You will never get a normal multiple choice question.
No. I was gonna do the A, B for this one, but I was like, I didn't want to keep looking back. Which one is A? Which one is B?
No, valid. Better to just say it.
Yeah. Dude, what are you doing for Christmas?
Going home.
Nice.
I don't know. I'm not doing anything. I don't have anything planned. I might ski once with my dad, but that's about it.
Oh, yeah, Vermont's good for that.
Yes, it is. If it snows.
Oh, dude, surely it's gonna snow. We're gonna hit with.
snow for the last 4 Christmases.
We're getting hit with like an insane storm this coming week, like this weekend.
No. I couldn't have been last week.
Why, cancel your classes?
Yeah.
Yeah.
Dang it. Wait, here?
I don't
Or
know.
in
I mean,
Boston?
just like Mass. No, Boston doesn't get snow that much because it's like right next to the water.
Oh, yeah, true.
But like, yeah, Mass. Maybe Vermont gets it. I don't know.
Probably southern Vermont.
Yeah.
Northern Vermont never really gets it.
Really?
When we get some, but we're like in a valley, so like all of our weather is like different from the rest
That's
of
true.
Vermont. So,
That's true.
What are you doing for Christmas?
We're going on a cruise.
A cruise?
Yeah.
Whoa.
Yeah.
Where?
In Miami.
To
Oh,
anywhere?
sorry. To the Bahamas and then back to Miami.
Oh, that's very
Yeah.
exciting. That's cool. Is it like a themed one or just like a normal one?
It's a normal one.
Okay, good.
Wait, what are the themed
Disney
ones?
cruise.
Oh.
Show the Disney adult.
All right, you guys ready for the answers? Who wants to shout them out? Number 1. Yeah, 5 to 3. Number 2. Yep, 3 to 5. Number 3. Yep. Number 4.
Relative to the replication fork, there's not relative.
Yeah.
Means like, what
Oh.
what does that mean?
Oh, sorry.
What
So
does
like
that
yeah, I
mean?
mean if you have the replication fork here,
Yeah.
and I mean either way technically, whichever way the lagging strand is going,
Okay.
it's gonna move 3 to 5. Does that make sense to everybody? Yeah. The Okazaki fragments will be synthesized 5 to 3 as the whole fork moves 3 to 5 for that lagging strand. What about leading strand? Or did you have a question?
Yeah, no, I just don't get the relative to the replication fork.
It's like, do we have a diagram like that?
You said?
Yeah, the lagging strand would be 3 to 5. I'm gonna see if we have a diagram earlier on somewhere.
Like, are we talking relative to like the template strand?
Yeah.
Okay.
Yeah, relative to the template strand. Maybe I should have said that. But yeah, like, oh, thanks. But yeah, both of these lagging strands, they are going, how do I say this? Yeah, so like if this is the 5 strand, or the 5' end, this is the 3' end of the lagging strand, and it's coming this way, so it's coming 3 to 5 of the lagging strand. But for the template strand, it's going, I mean, this is also a template strand. I mean, for this template strand, it's going 5 to 3. But like the direction of this fork is going 3 on the lagging strand to 5 on the lagging strand. Same thing here. The lagging strand 3' end is here, and the 5' end is there, so it's going 3 to 5. Oh, got switched. Yeah, and then leading strand is 5 to 3. Okazaki is 5 to 3. RNA synthesis, 5 to 3. I put that in there intentionally to throw you off, just to conceptualize like which direction the actual strand is moving. But it's synthesized always 5 to 3. Yeah, cool.
The third question is DNA is read. I think it's in both directions. Because like the DNA template, they just read.
It's like, here, I'll go back to that thing. It can, like, yeah, it moves in one entire direction, and so you might think it's going both, but it's being read this way too. Like, so like if the synthesis happens 5 to 3, it's happening this. It's also being read in increments 3 to 5. So it's being this strand is being read this way.
I don't really understand that.
I like to think about it like for that question. I like to think about it as it's like relative to the fork, like the direction of the fork. Because like if the fork is opening and moving to the right, the leading strand is going 5 to 3, and the lagging strand is going 3 to 5.
Yeah.
It's like if you're looking at it, like if you're reading it in the way that it's being like in the direction of the fork, the leading strand is going to be 5 to 3, because the 3' end is at like an end on the right side, and then for the lagging strand, the 5' end is going to be on the end on the right side, and like same thing goes for the left. You can see that they're like essentially like going the same way, like 5 to 3 for lagging, or 3 to 5 for lagging, and then 5 to 3 for leading. If you're looking at it the way that the fork is opening, not like how it's being read or synthesized. Like when it said like relative to the fork, I was thinking of like the way that it opens.
Yeah, that was the kind of intention with that too. Yeah. Okay. Cool. I think that was all the questions we had.
Yes.
Anybody have final questions? Yeah.
For the first practice multiple choice one.
Of this last two?
The first of the multiple choice. No, sorry, no. The first of the practice multiple choices at the end. So the one about the different phases. Yes. With the ORC is phosphorylated. I'm agreeing that it would be phosphorylated in S phase, but Is it not also phosphorylated earlier, or?
Not earlier, but it could technically be phosphorylated in other phases, I guess. Like G2, stuff like that.
I thought it was late.
G1, it needs to be not phosphorylated so that the pre-RC can form. Yeah. Oops. Okay. Yeah. So just general tips for the exam. Take your time. Take breaks. The exam is 2 hours, but we know how like Laura's pretty lenient with time, so we're not going to cut you off at 2 hours. So don't rush through it. Take your time on that. Ask us questions. I will be there tomorrow, and so will Esther and Maggie, I think. So please ask us questions. We want to help you. We will help you through your thinking. We can't give you direct answers, but like sometimes just talking to us can help. I said this last year too, and it came true. Laura loves asking about what proteins are enzymes, what proteins are like GTPases, ATPases, stuff like that. So make sure you know that because she loves to throw that in the multiple choice questions. And yeah, that's all I got. Good luck tomorrow. Thanks for coming. Stick around if you have any questions. Yeah.
Can you remind me of like the one of the enzymes you were just saying? She always talks about she likes to test on it and everyone gets it wrong whether it
Ran?
works to an enzyme or not.
Ran and RAS?
Oh no, gaps and gaps, right?
Oh, gaps and gaps.
Yeah.
So the gaps are
Oh wait, shit. Which one is it?
Gap and gap.
Yeah, gap
Yeah.
and gap are not enzymes.
Yeah.
Yes, they help the GTPase. They help the GTPase perform its enzymatic function, but gap and gap themselves are not enzymes.
Their
Yeah.
name is GTPase activating protein, so it has protein in the name, and the other one's guanine exclusion factor.
Exchange factor.
Exchange factor.
Yeah. Oh yeah, and also pick up quizzes and stuff if you didn't
Yep.
do that already.
So I have a question.
Yes.
So Thank you.
Yep.
Yep.
I don't know if I'll use it, but I feel like this exam's gonna be long. I have accommodations to get up and take breaks. Do you want me to come up and tell you, like, hey, I'm just gonna walk around? Or do you want me to just, like, get up quietly?
That's up to you, I think. Yeah. I mean, I won't care. If you
Yeah,
just
I don't
leave.
think we're keeping track, so.
Yeah. But I don't know. If
See you, Ricky. Thanks for
Laura
coming.
already knows, I assume, right? Yeah. So you should be fine.
Okay.
Yeah. Oh, people are just.
Oh yeah, here. Let me.
What's up?
So, I just have questions regarding some of them. I generally don't understand why or how I lost the points. I've been wanting to ask these for a minute now,
Okay.
but I didn't
Sure.
get the chance to. For this one, I do not understand how I lose 1.5 points for not mentioning proteins, even though the question doesn't say mention proteins.
Topic 9? I misread, yeah.
I'll just pull up
The first question, I'm just guessing I misread. This is asking.
Okay. I think they just wanted more of an explanation. So like the answer is yes, so I think you should get a full point for getting the right answer.
Because I lost a full point for getting this one wrong with my previous questions, but I don't get anything for saying yes to this.
Yeah, I think you should get a full point for yes. And then, but the whole idea of explain was like it's already telling you that it's phosphorylation and dephosphorylation, so you're kind of just repeating it when you're saying it's activated by phosphorylation, deactivated by dephosphorylation. The idea here is that she wanted you to mention WEE1 and CAK, CDK activating kinase. But I still think you should get a full point for like saying yes and like yeah. Was there something else too?
Yeah. These two questions, I just don't understand why I got zeros on them.
Okay. Why did you get a 0 for this? I don't know. This is right.
Right? I was tripping. I was like, yo.
Okay.
Oh, okay, I see. It's because you said they're present. Like it happens when they're not present, but it still doesn't happen during S phase. There's a normal occurrence. Yeah, and it does also have an early G1, but your answer is still right. It's not a 0. It's maybe like, yeah, the explanation is a little wrong, but the answer is still no because it's not happening during S phase. So I'd ask about this, ask about this too, and then this one too.
Mm-hmm.
What did she want for this? Exam 2.
So I have to take this to her and
Yeah, you would have to. She might be in the office right now if you want to go see.
that'd be great. The office is nearby, right?
Yeah.
Straight down the hall, you take a right, and then you're gonna take a left. It's gonna be on your left hand side.
Thank you.
Yeah, this also should not be a 0 because the answer is yes. What?
What about the explanation? I mean,
Yeah,
since
I'm
it
looking
doesn't
for the
have any
explanation.
explanations on there, I don't know what I got wrong. Is the
Yeah, I
thing.
don't understand this. Uniform. Positive and negative effects. Okay, I would say that only here you're only talking about negative effects on replication initiation here. But again, that should not be a 0. This should be at least like 1 or 1.5 points, not a 0. You're missing talking about positive effects on replication initiation. But you're not. But the answer is still yes, so that's a point. And you gave a good explanation for negative, so that should be another 0.5. So I would say 1.5 for this, 1 for this, and 1 for this. So yeah, I would bring that to Laura because I don't think the grading was right there.
Okay. If you have a question to ask, you can
Oh,
go ahead.
if you're not done, you can I don't.
Oh.
I think I just this one more, just this one.
Okay.
I'm pretty sure I got a point for the graph and I got a point for the explanation, but in total, whoever did this, I think gave me. Can you just?
Yeah.
Yeah. So I mean, think about like basic decision repair is like if you lost a nucleotide in ATPC, or if you can uracil, right? So like what happens is first that uracil is taken out, becomes just the sugar and phosphate, and then that sugar and phosphate is taken out and replaced with an entire nucleotide. Okay. So it's like basic decision repair is kind of like nucleotide decision repair, except it has a couple more steps in the beginning.
Oh, what are the steps?
Oh, I guess I can pull it up. Yeah, sorry.
That's okay.
Yeah, so
What's the actual answer?
yeah, right here. So you see the U? That's so first of all, the U gets taken out.
Right.
Right. Then, sorry, your head's in the way. You're good. So then, once the U is taken out, then the entire like sugar phosphate, the backbone part, that's also taken out, right? And then added on. Nucleotide excision repair. Think about it like.
Okay. The idea here is that like it's not going to shrink after.
So nucleotide excision repair is
Like it'll just stay plateaued because
it
like
works the
that
same way
thing is
in that
stuck at the end, so you're just not going to get
but, like,
polymerization or
instead of, like,
depolymerization.
If this is the problematic dimer, it's
But
not just
wouldn't
like
they
those two things.
depolymerize
There's a whole
if
chunk
you
that
leave
gets
them
replaced. So,
there
like,
long enough?
There's nicks.
Because I remember asking this to the
and
TA
they just
that
take out the
was
entire strand.
proctoring the exam.
Because this, the
I asked them,
dimer here that
am I
causes
supposed to
confusion
think
or
of this
whatever,
like a long period?
It's problematic not
Because
just for
eventually
itself,
it's
but
gonna
for the area around it.
Yeah.
break
they
down
can take it off.
regardless.
Right. And then
Yeah.
add it back.
I mean, there's no
Like,
way
the
it's
new
gonna
ones get that
stay,
back on.
together
So
for
it only
for
replaces
eternity. Like,
the strand
Yeah, that's
with
true.
the error
Especially,
on
does it
Yes.
say that there's a?
Okay. But like the bottom, because wait, this is the one that it's the pyrimidine dimers, right? So
yes.
that's. But is
Okay,
that a
I don't-
double strand?
which is why
I think
also,
in a long period it would still
It
stay
can
together
lead to a
because
double-strand
in
problem.
the
It's not fixed by
like
this.
Okay, no, that's only called
directions it
You
says
want me to
there's a
go to
microtubule
No, it's okay.
organization
I
here.
understand
So it'll
that.
have
Okay, the
cap to minus
non-homologous
n and a cap to plus
again,
n,
so
so it'll
yeah.
just kind of stay stable.
Um,
So it won't degrade.
Okay, and do you think? Well, we don't really cover it much like in the
but yeah,
lectures, but do you
I
think
still
she'll
think
ask
this is
about the
like-
details of
Not
homology directed
maybe
repair?
like
Because
half points at
she just
least.
kind of like
Yeah, so I would-
we didn't
Okay,
really go over
I'll-
in the lectures and
you
Just
said
know that
she's in
it
the office?
requires a
She
template.
should be right now,
Right.
maybe. Yeah.
That's kinda
Sorry
convenient.
about that.
Okay.
Yeah. And then non-homologous is like
Yep.
you don't need a template, so it happens faster.
What's good?
Yeah.
But you also lose information.
Yeah, that one we went into like a little bit more detail.
Yeah.
Okay.
So yeah, just know that for HDR, it's you have a separate template that comes in and fixes it exactly.
Okay. And then the last thing, I'm sorry to ask again,
So
but
with
can
the
you
replication
explain
work and like the
the,
bubble Okazaki
I
fight?
forget what topic it was,
I'm
but
trying
with
to, like,
the
find a
two
good
strands, and then it was like
way,
A and B, and then there was like the origin,
should I kinda,
and then it was asking which
focus
one would be the
on,
leading and lagging strand. Can
Yes.
you go back
Yeah,
to
yeah,
that
yeah, absolutely.
picture?
Yeah.
on
That was this
Yeah,
one. Yeah.
Wait, let me think. Yeah. Okay, so A is the lagging strand.
So think about So we'll start like from before that, right? So origin's here, right?
Right.
You draw the bubble like this, right? And so the lagging strand is moving 3 to 5, right? So just think moving, like which direction it's going to eventually end up in, right? So if this is the template, like this is 5, starting 5 here, and then that's 3 over there.
Right.
Right. So then under it's 3 to 5.
Right. That's
So like if you kind of put a line right there,
I mean, as long
that's
as
the
you
lagging
have
strand that gets
my
made,
grade and
which is getting
everything
made
to put in there,
3 to
I
5.
could check, like, still have it
Right?
back,
It's actually getting
I
made 5
also
to
have
3,
the
but
answer.
but like it's
The
moving
grade,
3 to 5
okay, that's
Yeah.
perfect.
to be made, right?
Yeah.
One last.
So
question.
because sorry.
What would you
So
recommend we study
because
for
it's
the
moving
exam?
overall
The
3 to
quizzes,
5,
the exams,
it needs to
or
be
the
made in
problem
small
sets?
chunks
All
of
of
5
it.
to 3.
All of them?
Right.
Which is the fragments, right? So then
100%
replication of this strand involves
Isn't
separate
that too
separate
much?
prime molecules,
I
several
mean,
prime
okay, if you
molecules,
think of it this way, right?
which you need a
Your
lot of primers
exams
for the a lot of
It's like a
fragments that are
very
made.
good like replication
So
question.
B is
of
the
what was
leading
given on the
strand.
problem set.
Yeah.
But
The problem set's like a
the strand
maybe
that it
like a
produces is 3 to
smaller
5.
version of an exam
B.
question. Your quizzes
Yes.
are also
So B
smaller
is 3 to
versions of the
5
exam question,
if you're looking left to
but they
right.
all
Oh,
let
okay,
you
so-
think
So it's gonna make
it's
a
just
5 to
different
3 left to
variation of
right.
the exact
So
same question.
the
So I
5.
say everything.
Okay, so the 5 the
Yeah.
the strand that it
I mean,
produces
he's TA'd for
5
two
to
of
3.
those
That's the
classes,
leading
too, so
strand.
he knows what he's talking
Yeah,
about.
wait, let me see if I can bring this
Also,
thing.
narrow problem studies.
Yeah.
Yeah, I guess.
Yeah. Good luck.
Thank you. What's the
348. So if you go down the hallway, turn right and turn left, you should be there.
Yeah. So
So
yeah, so this is the leading strand template, right? Because the leading strand is actually this.
Right.
Yeah. Which is made
Right.
this way, right?
Yeah.
This, if that's the 5 end, this is the 3 end of the template.
The leading strand.
Yeah, the leading strand. Leading template, right?
Yeah, that's what I'm just confused about. Like, I'm gonna definitely forget about it because like I just like
Just think of it in like rules, right?
because I always think of the leading strand as being
So you
like 5
read
to
the-
3, which like it
these are
is
what
there,
conditions,
but
right?
like
The
it's not.
flags.
Like I don't know what it is.
The way, yeah, no, it's fine.
Like, and I got this question wrong on like the quiz. too.
Correct.
Okay, so so like, I'll just draw it again, right? So these are the parent, right? So P.
Well, it'll be
Actually, that's good.
made
Say
5
P,
but it'll be
parent
red
strand, right?
5. So the template.
Oh yeah, I'm gonna read the template.
And then let's say the origin happened
but
here,
when
right?
you synthesize, it's gonna be like
So,
the 3 is
that
gonna
bubble
have
that gets
5
made is
prime.
kind of like that.
Right.
And
So,
then where your 5 prime end on
The
template
if we're
strand,
labeling
you
strands,
synthesize
right,
it.
and we already know that this is the
Yeah,
3
it's
end
gonna
of
go
the parent
straight,
strand,
right. So
that means we
means
know this is the 5 end of the parent strand
separate
down here.
strands, so you
Mhm.
just
If
move
that's the
it
5
through
end, this is the
how it's supposed to be. Yeah.
not the 5 end, so
Right.
3 end,
Like,
right?
it shouldn't be a worry about
And if this is the
whether
3 end,
mess
this
it
is
up.
the
So
The 5 end.
5 end, right?
always remember that my
Of the
leading
template.
strand's
Of the
gonna be
template. These are all template, right? Because this is just the original
in
problem.
the-
Now, the new strands that get made,
Synthesize 5 to 3, but when I read it, my leading strand is gonna be
I'm not
starting
like calling one lagging leading yet,
Yeah, so if you
right?
see-
So we'll just
If you
label
see a
them.
5 prime
Like they're
end,
opposite.
like on
They're
the
opposite,
end
right?
of
So
your
then
template strand,
this
so if
end
you
of
have a
the
diagram
new
like
strand
this,
would be
What you like only
5.
give
5.
the like two
So I'm just
templates.
gonna
Yeah,
make
if
an
you
underline
give
for the
two
new.
template strands,
And then this end would be the
whichever end
three.
has
Yep,
5
and
prime
then
on
that's
the
the
end,
opposite.
Opposite,
it's
right?
gonna
So
be
year
the
5
strand that
and
the leading strand is synthesized off.
so
Okay.
Right. And so when you
we
have
know
like
that this is the
I always find
origin,
it
so it has to move away from the origin,
Mhm.
right? So if our direction is going that way, which way can be made first? Or sorry, which way can be made without interruption? The one that's already going 5 to 3, which I define as this one. 5 this way to 3.
Right.
Right, so leading.
Knowing where I'm
This
gonna
one,
be putting my,
it's going 3 to 5.
my lagging
So
strand
it
on
needs to be
the- so my
synthesized,
leading, I
like
know it's gonna be
molecularly,
3 to
5 to 3.
So my lagging
So it'll be
is
synthesized
a
in this
lagging
direction.
Mhm.
Does that
One.
make sense? From
Um,
the-
Two.
because
Like it'll
this
synthesize
It goes the
these
opposite way
little
from the fork,
fragments,
yes.
right?
It goes the opposite way from the
That's the way I remember. I don't try to like memorize, you know, if it's top this,
So
bottom
we have
that.
to read our DNA.
I'll just draw out my steps
and we have to
and kind of
synthesize
work my way through it.
Okay.
Yeah.
Our prime
If that sort of makes a little bit more conceptual
Yeah,
sense.
it does, but I mean, I'm definitely gonna forget about it. I'm so tired, so. But it's okay. I mean, I'll try not to. Can I take a picture?
Yeah,
of
go
that?
for it. Yeah, you want me to turn off the screen?
I think let me see if I'll.
Cumulatively like move in the direction of the force.
I'm gonna go find an eraser to get up.
So, this lag exchange, right? That's not the lag exchange for this though, correct?
What do you mean?
So
Yeah, the picture came out fine, so like I'll just copy this down.
Yeah, I was gonna say if you want the whole thing without all this random stuff on here.
Oh yeah.
Okay.
I mean, what you wrote is like pretty much everything, so I think it should be fine. I mean, yeah, I'll just copy this down. I mean, this is like the thing I'm mainly confused about.
Mhm.
I'm confused about some stuff from topics 1 to 6 because the review ran short of time.
Oh, really?
yesterday,
Okay.
So we didn't get to cover everything.
Okay, what did you miss? I think you can go over stuff if you
6.
want.
Well, like a lot. No, it's okay. I'm just
Okay.
gonna rewatch the lectures, but yeah. Well, actually, I did have one question. Maybe you can answer. It was
I know on the, I think it was.
What was topic 6 again?
I don't even remember if my question was about topic 6.
Oh, okay.
Topic 6, we didn't even get to it like at all.
Oh, really? Okay.
We only we rushed through topic 5, but we didn't even cover it. I think topic 5 was like the like all the pathways.
Yep. Yeah.
And then 6 was like microtubules and stuff.
Oh, I see. Yep.
Yeah, but then wait, I'm trying to remember what my question was.
Yeah, sure. I'll let you.
I don't know if I didn't write it down. I should have written it down.
So I was supposed to like draw in where I would I know it's kind of and
Oh, okay. So I don't know how much you know about the tryptophan, like operon.
Yeah, I can.
Okay, but it was my question.
Because I know I want to.
Okay, so so our tryptophan. Okay, so if there is low tryptophan in the environment around the cell, will that have the same effect as there being low tryptophan tRNA in the cell?
Yeah, because if there's low tryptophan, there will be low tryptophan.
The difference is just that tryptophan itself represses the it binds the or it's the co-repressor, whereas tryptophan tRNA, the codons are the tryptophan codons.
I see what you're saying. So are you saying that like if there happens to be low tryptophan but there happens to be high tryptRNA?
Yeah.
That's like an unlucky situation, right? Because the cell reads the tryptRNA saying, like, oh, okay, we have stuff, we don't need to make more, right? But there actually ends up being less. So that's a problem. But like that's like a situation that can happen, doesn't happen very often.
Right.
You have to split down.
So
everything.
Well, wait. Okay. So when tryptophan tRNA is present, the ribosome can translate through the codons. And then, this is my other question. Do you know if it stops on region 1 or region 2?
To make the
Okay.
it stops
Now
on
it's
2.
clicking.
It stops on 2, which
She
blocks
explained
the
this to me like that,
tubule
like how
from
it's
forming,
like-
Which makes 3 and 4 to
But-
form.
Right, which ends
which attenuates.
you're right.
Yeah.
So like just thinking about
But
it,
then
it
tryptophan
still needs to
being
go in the
present
5 to 3
stops
direction,
it, like,
because
before
you
that.
start
On
from the origin.
Yes.
You're
Stops the
working
ribosome
backwards.
on region 1.
Yeah, because then the 2-3 forms.
You're working backwards,
which makes the
synthesizing
3-4
in
not be
opposite
able to
direction to the 4.
That continues.
So you can still keep going
So you
5
can,
to 3.
because it's connected to
continues
this. So this
transcription?
is just going constantly this way.
Yeah.
So this is like connected.
But
here.
why would I thought
So this is
transcription
also
only continued if
basically being
tryptophan
dragged
wasn't
along this way.
present?
So sorry. I
So
had the
everything's
wrong
moving
one.
this
So
direction,
if it is
but
present, it'll
because
stop it
can't
on the
synthesize
second
in that
one.
direction. It has to synthesize in this
If
direction.
tryptophan is present, it will stop it on the
do this,
region 2.
Yeah.
and then lift off,
Making the 3-4
and then do
hairpin.
this.
But then what's
Stop.
the difference between that and
So that it's
tryptophan
still
tRNA?
going.
Yeah, so
So
your
it's still being
fragments-
synthesized 5 to 3.
When is
Yes. So your
tryptophan
fragments
present?
that are
When
closest
there's
to
no
the center
tryptophan in the
will
environment?
synthesize
No,
first.
so it's like
One,
it's like
two.
if you have like
Okay,
a lot of tryptophan, it'll start to bind to its tRNA and make tryptRNA, right?
Okay.
And so I'll talk about like if there's a lot of tryptophan in the cell, there's going to be a lot of tryptRNA, but it's irrelevant because cell has a different
Well,
mechanism,
I
right?
thought-
That has the inhibitor, right?
Right.
Right. When there's a moderate amount, the
Again, I think it's really helpful
that
to
to a
just
double
draw
codon that gets
either
translated.
Mhm.
So you can just
And
put that here, right? So
let me refresh myself. I'll pull up their slides.
Okay. Because what I thought was that the tryptophan tRNA was only like present and like working when the cell thought that it had to produce more tryptophan. So that would be the situation where the ribosome would translate through those tryptophan codons.
I think it's on topic. You said it was topic 4, right?
Yeah.
Yeah.
Perfect. Yeah.
No, this only makes sense if you
Yeah.
I understand how we can build off, but
So,
This is your
Um,
5, so this is what says 5 and 3.
so over here, yeah, let me pull down.
This is why it has to go in that direction. Like, build in that
So,
direction. Like, as a whole.
this peptide, they're talking about this one right here,
Mm-hmm.
is, maybe back up just a second. Should probably back up a little bit.
so beginning of my arrow is gonna be at the 5 direction, going towards the 5 prime direction.
Right, so
Of the template string.
if there's high levels of tryptophan, transcription won't
so-
even happen, right? That's the first
So this
point, right?
is needs to be
Or
active.
above the first point.
I read the
And then
Right,
if
that's when it
there's
binds to the repressor.
exactly. So
Like
when there's a
how the
medium
prime
amount of
goes through the
tryptophan, there's not
bottom.
enough that binds to the repressor.
Oh, it's in this one also.
This is when we get into attenuation.
So overthinking was wording.
It's just,
There's like
it's
a-
a mess to get your head around.
There's
for
like a
sure.
sequence of words I'm trying to figure out.
Translating between pictures and words for it is also just weird.
Yeah.
It's like, yeah.
So that's
Yeah.
okay. And
Says
then
if there's enough tryptophan
This is
tRNA
the
around,
same
then the
thing.
short transcript is made. Does that mean that those two codons are translated by the ribosome?
when I hear several prime molecules, I
Yes.
just always
Okay.
think of Kozaki
Yes.
fragments because
Yes.
they're in fragments and you want several of them.
Yeah. You need a primer to start every Okazaki fragment. Do you need chip opera question?
Yeah, I'm just I have the understanding. I'm just not able to form in words.
Oh, is this with the hairpin?
Yeah.
Hairpin? What was the question?
I was just, well, I was confused.
Ah, here it
about-
is. Here it
oh,
is.
okay.
Sorry. So the medium, tryptophan is separate from tryptRNA because tryptRNA is used
Where are you going?
My replication.
to make more tryptophan or make more protein, right?
Mhm.
So those are two separate entities,
Right.
which is my confusion. I was telling you that they're like one and the same,
The effects are the same, but they're not.
right?
Because there's no 3 prime.
So that tryptRNA, which is made separately, that has the
lagging
fine
strand, also the
regulation.
strand with Okazaki fragments.
So basically, when protein needs to be made or tryptophan needs to
Well,
be made,
just can't be filled
The primer gets removed and the primer.
the tryptRNA is active. It gets used. It gets degraded.
the primer that we
Right.
need to have Okazaki fragments.
In that medium
Yes, 'cause
tryptophan
every Okazaki fragment needs a
concentration,
primer, right?
not all of it's
And so
degraded yet.
On the
Mhm.
very
Right,
last
so
Okazaki
either
fragment,
There's
on
enough of it to
the
go,
DNA,
enough of it to be
that last
sensed,
primer gets
saying like, hey,
removed,
we don't need to make
but
more.
there's nothing at the end of the DNA.
Or there's
strand,
not
right?
enough being
So you
sensed,
can't
saying,
synthesize
hey,
off
we need
of
to make
that.
more.
So telomerase comes in, extends the
That's
template
kind of the best
strand so you can
explanation.
add another primer
Okay.
and
About
then
it.
fill that back in.
Okay.
How
Do
does
you
it
think Laura's
extend
still in
the
the,
template strand? Do I need to know exactly how
the
it
office?
dealt
Just
with
know
this?
that
Could be.
it
Okay,
it
I'm gonna
has
check.
an
Okay.
RNA template like within
Clarify,
itself.
'cause I like lost
and
my
it's
exam, my
builds
first
off of
exam,
that.
so I need to
The
remember if I got it
telomerase.
right or not.
Yeah.
Did she post the exam online or?
I think she posted the answer key.
Okay.
I guess I
Oh,
could look
but I
it
guess
back,
you were
but
looking
I
for
lost
your own.
mine, so I don't even know. Like maybe I understood it at the time, but I don't anymore. So.
Okay.
I mean, I get like the hairpins, like why they form and stuff, but it's just the tRNA that like I don't really like get it. But I mean I get it, but like I don't know, it's just a lot to like keep track of.
Bring in the
So
Yeah, sorry, I wasn't able to give you like the most clear.
no, it's okay.
What?
Yeah, wait. I have tryptophan?
Yeah,
I
TRNA
don't
like
move through.
skirts along.
Yeah, I guess I was just confused like where the ribosome pause is. Because like I said, we're kind of like rushing through it yesterday, so I don't even.
Continue if it wants to stay.
What did you say?
Because I wrote down that
So,
when tryptophan tRNA is
I
not
understand why
present,
tRNA
the ribosome will pause at region 1